adrenaline, right? The fight or flight hormone. So, how do we regulate, right? How do we regulate protein kinase A so it's only on when we need it to be on? And then, how does protein kinase A regulate downstream genes that are responsible for metabolism under these conditions, right? Either we're starving or we're responding to fight or flight, okay? Now, in addition to responding to different environmental conditions, again, starvation, if we've just eaten, right? We also need to regulate the activity of certain enzymes in a tissue-specific way, right? As we go journey through metabolism, we'll see that certain enzymes catalyze the same reaction, but they do it differently in different tissues, right? One thing that we're going to see very clearly throughout the semester is that the liver is its own separate thing, right? The point of the liver is to keep the rest of the organism happy, right? You can't live without the liver. And so a lot of the enzymes that are in the liver, though they catalyze the same reactions that we see elsewhere in the body, they have to do so differently for different reasons, responding to different stimuli. So how do we regulate when a protein is turned on in response to, say, insulin or some other hormone? And how do we regulate when an enzyme is active in specific tissues? We're going to look at those today. And we can control the activity of enzymes in 5 basic ways, right? I'm going to get number 5 right off the list here because we're not going to cover number 5 today, and it's also kind of common sense, but we're not going to really talk about this semester, which is simply just controlling the amount of the enzyme present, right? So by controlling the transcriptional activity of the gene that encodes for that enzyme, now we either have more of that enzyme so we can catalyze more reactions, or we can turn off transcription of that gene so we have less of that enzyme catalyzing fewer reactions. That's one way to regulate one way to regulate an enzyme, but that really has to do with transcriptional regulation, not so much regulating an enzyme itself. To regulate an enzyme itself, once it's been made, right, we have 4 options. We can have allosteric control. We've already seen an example of an allosteric effector, right? Does anybody remember what that is? We've seen an example of an allosteric regulator for a protein, specifically. For hemoglobin. All right, you got half the answer. Who else, right? So it does have to do with hemoglobin. 2,3-DPG, 2,3-bisphosphoglycerate. Remember, that's an allosteric inhibitor of hemoglobin, right? It stabilizes the T state so that hemoglobin binds less oxygen and can deliver more oxygen to the tissues. So we're going to go over allosteric control in a little bit more detail today. We can have alternate forms of the enzyme. These are called isozymes, right? Isozymes are important. This is how tissue level control works, right? So different tissues express different genes encoding for slightly different versions of the same protein. We'll see that later. Reversible covalent modifications are critical, especially in metabolism, for turning off certain pathways or turning on certain pathways. Okay, so we can modify certain residues in an enzyme itself, and that leads to some change that either activates it or inactivates it. And then lastly, we have irreversible proteolytic cleavage, right? Kind of contrary to what you might think, by degrading a protein or a pro-protein, a zymogen protein, by cutting that protein apart, we can actually activate it, right? So we synthesize a zymogen. Again, we'll talk about zymogens later on today, right? We synthesize this polypeptide that is inactive, it does nothing, and it's not until it's cleaved, right, that it becomes active, okay? So let's start first with allosteric control, right? And to do this, we're going to examine aspartate transcarbamylase, ATCase, right? And this essentially, we're going to see this again in chapter 23, or no, 26, chapter 26. This deals with the synthesis of our pyrimidine nucleotides, specifically CTP and UTP, okay? And how this works, again, we're going to cover the actual whole pathway later on in the semester, but carbamoyl phosphate reacts with an aspartate, the amino acid. We haven't talked about carbamoyl phosphate yet, that's okay. And it forms this intermediate here, okay, N-carbamoyl aspartate, right? So carbamoyl aspartate is the precursor for synthesis of our pyrimidine nucleotides, UTP and CTP. Here, what we're seeing is the committed step, right? So ATCase catalyzes the committed step in de novo pyrimidine biosynthesis. committed step. The committed step is the first step in a pathway that generates a product that is unique to that pathway, right? Now, because it has to generate a product that is unique to that pathway, right, it's generally the first irreversible reaction in that pathway. ATCase is allosterically controlled by CTP, the end product of this pathway. So notice, the product of ATCase is carbamoyl aspartate, but ATCase is regulated not by carbamoyl aspartate, but is regulated by CTP. Of course, it's going to be regulated by carbamoyl aspartate just due to the Le Chatelier's principle, but it's additionally regulated allosterically by CTP. Notice that the structure of CTP is very different than that of carbamoyl aspartate. They look nothing the same, right? So this can't be a case of just simple competitive inhibition with CTP binding or competing for binding of the active site of ATCase. It's got a totally different structure, so it's got a totally different shape. It shouldn't be able to fit in the active site. So the only explanation left is that CTP must be binding elsewhere on this protein, ATCase, and doing something to that ATCase to slow down the reaction. Okay, it's an allosteric inhibitor of ATCase, as we can see here. For any given concentration of carbamoyl aspartate and aspartate, so we're holding that constant, we can measure V naught, essentially the rate of carbamoyl phosphate synthesis, right? So this is V naught versus CTP. What we see is that V naught decreases. Okay, the velocity of the reaction decreases as we add more CTP. Right? How is this being done? Right? So let's start first with I don't know why you're listening today. Right? Let's start first with the committed step. Okay? What is the committed step? Right? This is going to be important to figure out as we look at more and more pathways. Right? So let's see which enzyme catalyzes the irreversible reactions. Okay? So each individual letter here is a substrate or product of some enzyme catalyzed reaction with the enzyme that catalyzes that reaction above the arrow, so E1, E2, and so on. Right? So which enzymes catalyze irreversible reactions?
4, 5, 7.
4, 5, 7. 1. Okay, good. Now, which enzyme is going to catalyze the committed step of this pathway? Kush-Patel. Where's Kush-Patel? Raise your hand. Oh, that's you? Huh? Kush. Yeah, just Kush. It says. Oh, now you. No Kush-Patel today? All right. Fine. How about Advic? Is there an Advic here today? All right. Tell us, what's the committed step? Huh? A to B. Okay. Why do you think A to B? Why are we saying that?
Because it's the first irreversible.
Correct. It is the first irreversible reaction. But what's the other characteristic of a committed step? It produces a unique product. So does E1 produce a unique product to that pathway? Well, it's producing B, and in our pathway, B yields C, but can B be used to do anything else? Huh? Yeah, we can turn B into B prime there. Okay, so yes, it is the first irreversible reaction, but it produces B, which is not a unique product. So we're going to see this actually when we discuss first glycolysis. There are steps that come before the committed step, because what's generated before the committed step in glycolysis can be used for other purposes in the cell. Okay? So let's go back to our example here. Which do we think then is the committed step? From C to D. All right, why? Right, it's irreversible, and D is only used in this pathway. Nothing else is happening to D, right? So when we're looking at a pathway, we're trying to see what is the committed step, right? Well, there's the first irreversible reaction that is generating a unique product. Now, why does the committed step have to be irreversible, right? If we see here, B yields C, which is catalyzed by E3, well, C here, okay, that's being used only in this pathway as well, in this example, right? So this is a unique product. Why is it that we have to look for an irreversible reaction?
Because it's not committed then.
Why is it not committed?
Because it's able to go back.
Exactly, right? If we say that our committed step here is E3, right? Well, yeah, we're generating C, which is unique to this pathway. But C, because that reaction is reversible, can be turned back into B, and B is not unique, right? So we're looking for the first non-reversible reaction because it generates the first product that can only be used in that pathway because it's irreversible, it can't go backwards, right? Now, why do we care about this committed step? Why is this committed step so important? We're talking about enzyme regulation, and I'm going on about this committed step in a pathway. Why do we care?
Is that the enzyme that you want to regulate or regulate the reaction?
Okay, that's the enzyme we want to regulate. Why is that the enzyme we want to regulate? Do it here. Okay? Why is that the enzyme we want to regulate? Huh? Partially, right? So we said, all right, the committed step enzyme, or the enzyme that catalyzes the committed step, that's what we want to regulate in a pathway. Why is that the enzyme we want to regulate in the pathway? Right, so if we're regulating the committed step, all right, or regulating the enzyme that catalyzes the committed step, now we regulate entry into that pathway. Everything else that comes before the committed step, we can use for other stuff. By regulating the committed step enzyme, now we can regulate entry into that committed step. And what we'll see is that the enzyme that catalyzes the committed step in almost every pathway is the most heavily regulated enzyme in that pathway. For example, phosphofructokinase, which is the enzyme that catalyzes the committed step in glycolysis, is regulated by several different effectors. All right, several different molecules regulate the speed of phosphofructokinase because that's how we regulate entry into glycolysis. Everything that comes before the phosphofructokinase step, we can either store or use for other purposes, but as soon as we generate fructose 1,6-bisphosphate in that step, okay, now we're committed to glycolysis, so we have nothing to do. So if we are unnecessarily entering glycolysis when we don't need energy, well, now we're just wasting fuel. And conversely, if we're not entering glycolysis when energy is low, well, now that's problems for ourselves, right? So we need to regulate these enzymes that catalyze the committed step. So to distinguish an allosterically controlled enzyme from a quote-unquote normal Michaelis-Menten enzyme, all right, let's remember what the plot for a normal enzyme looks like, right? So here, again, on the y-axis, we have what is functionally V naught, right? The rate of formation of carbamoyl aspartate. We get a hyperbolic plot, right, for a non-regulated enzyme, right? What sets an allosterically regulated enzyme apart, okay, is the sigmoidal nature of its velocity curve. So instead of hyperbolic, we get a sigmoid, similar to what we saw, at least in shape, to the hemoglobin loading curve, okay? So Michaelis-Menten kinetics are not observed in allosterically regulated enzymes. And because a large proportion, at least more than half, of all enzymes are allosterically regulated in some way, okay, we see this sigmoidal plot most of the time, right? What else leads to a sigmoidal plot? Right? So yes, allosteric regulation. What else leads to a sigmoidal velocity plot? What might? cooperativity, exactly, right? So cooperativity and allostery both lead to sigmoidal curves, right? We saw cooperativity with hemoglobin. That led to a sigmoidal, what we call the loading curve, but if we consider that the reaction, quote unquote, that hemoglobin is catalyzing is really just binding oxygen, we can functionally just replace fractional saturation on the y-axis with E naught, right? And that would lead to the same plot. So both allostery and cooperativity lead to a sigmoid there in the velocity curve. All right, so before we discuss how this sigmoidal curve comes about and why it comes about, let's look at the structure here of ATCase a little bit. So ATCase consists of two catalytic trimers that are stacked on top of each other, linked by three regulatory dimers. Right? So what are we talking about here? If we look at ATCase from the side, right, what we have is two layers, right? The first layer on top looks like this, right? So we're moving to the top bottom. We have a trimer of three catalytic subunits. And on the bottom, we have the same thing, just sandwiched below it, right? So we have a total of 6 catalytic subunits. Each catalytic subunit has its own active site, right? So an entire sandwich here has 6 active sites and can catalyze 6 reactions at a time. Now, each of these catalytic subunits are connected to a regulatory subunit, right? So here, this regulatory subunit is connected to the catalytic chain. Here, this one's connected to the one on the bottom, and so on and so on. So we have a total of 6 regulatory chains. And these regulatory chains form dimers, right? So we look at the bottom view there from the side, we see that we have a dimer here of regulatory units. Here a dimer, and behind we can't see it, another dimer. This is what's holding the entire quaternary structure of ATCase together. Now, significant contacts exist between the catalytic and regulatory subunits. We can see that there in the red, right? So if you finger here, we have significant contacts here. They're not covalently linked together. Instead, they're linked together, and the zinc ion there is important for mediating that contact between them. They're being held together by weak interactions. All right. Now, PALA, right? If we see here PALA, I'm not going to try to pronounce that. I'm just going to embarrass myself. I said I wasn't going to. PALA here, okay? PALA is what we call a bisubstrate regulator, a bisubstrate inhibitor. What this means is that it's simulating what the transition state looks like between two substrates. So it's a bisubstrate inhibitor. And what we see, right, if we look at our bounds, at our substrates, which are both going to be bound by the catalytic subunits, right, we get our carbonoyl aspartate, or carbonoyl phosphate rather here, our aspartate here, and we reach this reaction intermediate, right, so this transition state, before we actually generate carbonoyl aspartate. Remember, all the way back here. Okay, so we reach this transition state, this intermediate first. Now, if you look at the intermediate structure and the structure of PALA, you'll see that they are very similar, right? So similar, in fact, that PALA strongly binds the active site, more strongly than either carbonoyl phosphate or aspartate, right? And why is that? Why does PALA bind stronger than either carbonoyl phosphate or aspartate?
Enzymes bind like stronger to the intermediate subreactions.
Exactly. Enzymes have maximal binding energy to not the substrate, but what? The intermediate or the transition state. Okay, so by synthesizing this molecule, which resembles that transition state or intermediate, now we get stronger binding. It outcompetes. The KB is much smaller. Right? It outcompetes both carbonoyl phosphate and aspartate for binding of the active site. And what does that mean? Well, now that means that carbonoyl phosphate and aspartate can now no longer enter the active site, so it's an inhibitor. Okay? But when PALA is bound to ATCase, we can say that that is resembling what it would look like when the substrate is bound. Okay? Even though it's not going to catalyze the reaction, okay, but it resembles what it would look like when these two are bound. So it's functionally what it would look like if ATCase bound carbonoyl phosphate and aspartate, formed the transition state, and then was frozen in time. Right? So we can take a snapshot of what this looks like. Right? Let's see. All right. So let's see here. Another question. Right? Histidine 134 in the active site of ATCase catalytic chains, right, which you can see up there, is believed to be important in stabilizing the transition state between of the the effect will be of pH on the rate of carbamoyl aspartate formation. So if the pH is high, right, will the reaction rate increase or decrease? If the pH is low, will the reaction rate increase or decrease? What's the pH dependence? Is there a pH dependence? Here we have, this is the 134 over here. Here we have the intermediate, right? This is the transition state. Right, so you see the binding between the active site and the transition state. So what do we think would happen as pH increasing? Would anything happen? Would the velocity increase or decrease? Advik here, or Advik, Advik, huh? Oh wait, did I already call it? Yeah. Oh, okay, sorry. Sachin or Sachin. Let's say the pH increased. If the pH increased, what would happen to velocity? Would decrease. Why do we think it would decrease? Correct. We would be in a more basic environment, yep. So a more basic environment would affect the interaction between the amino acids. All right. Would that matter? Sure.
I would say when the pH increases, the reaction rate would go up because I think things need bonding.
Because the histidine is what?
Hydrogen bonding.
Right, so this histidine is contributing a hydrogen bond to the intermediate here, right? Let's say to the transition state.
So when the pH increases, the pKa of the histidine rises.
Right?
So if the pH increases, it's gonna keep more hydrogen.
What happens when the pH is higher than the pKa?
Lose it.
We lose the proton.
Oh, so it goes down.
So it goes down, exactly, right? If we're decreasing the amount of interactions that we can form between the active site and the transition state, well, that decreases the binding energy, makes it less stable, so the activation energy effectively goes up, right? So by increasing the pH and losing that proton, histidine 134, well, now we form one less interaction, right? The binding energy goes down, activation energy goes up, so rate goes down. And conversely, at low pH, right, that we keep that proton, the reaction rate would go up until we denature the protein, right? What happens in our stomach? Our stomach has a pH of about 1, right? Why is the pH of the stomach about 1? We haven't talked about this, but who knows? Why is the pH of the stomach so low? The same thing that happens when the pH is too high. There's a serial killer in London about 100 years ago, and he would befriend old widows that had a lot of money, and eventually they leave him all his money, and he became very wealthy, and they couldn't figure out what was happening to all these widows. Turns out what he was doing was he was killing them and then dissolving them in lye, which is sodium hydroxide, and dumping them out in the backyard. And the way they found them was that they found all their false teeth. The reason why you do that, right, is that both high pH and low pH denatures all sorts of biological material. Denatures proteins, it digests lipids and cell membranes and all these other kind of things. So it just turns into soup. That's why the pH in our stomach is so low, right? As opposed to being high, we function the same, but that's not how our bodies work. By pumping protons into the lumen of the stomach, right? Well, now we denature all of our dietary proteins and all this right? So the same thing would happen here. If the pH were too low, yeah, we would get that proton back at the risk of denaturing the entire protein.
Is that proton is that particular proton acidic? Because
Which proton? This one here?
yeah, so that proton is
Well, this has got, it's got a pK of about 6, give or take a bit.
that's where the one the nitrogen that's for the other
What other nitrogen?
the 1 year to the
Well, remember, the imidazole ring exists in a resonance structure, so it could be either one. The imidazole ring of histidine exists in a resonance structure, so that proton is in the ring or not in the ring, which is that nitrogen is. Right? Okay, now ATCase exists in a T or R state, and we've seen this before, of course, with hemoglobin, okay? And the same is true of all allosterically controlled enzymes, right? They exist in either a T state or an R state. Now, the T state and R state can be affected both by cooperativity, like we saw with hemoglobin, and with ATCase, okay? And it can be affected, the equilibrium between the T state and the R state can also be affected by whether an allosteric regulator is bound or not, right? Now, in this case, ATCase does bind, of course, its two substrates, carbon monophosphate and aspartate, and like hemoglobin, right, binding the first set of substrates makes it easier for the second set of substrates to bind, which makes it easier for the third and the fourth and the fifth and the sixth. So it exhibits positive cooperativity. Now, when we switch from the T state to the R state, okay, the two catalytic trimers move about 12 angstroms apart from each other, and they twist about 10 degrees away from each other, right? So they pull up and twist, so it makes them more open, right? So we have the T state on the left, which is less active, and the R state on the right, which is more active. This is typically how the T and the R state are defined. The T state is the state in which we have an unbound enzyme that is inactive, and the R state is where we have a bound enzyme that is active. When cala binds, because it binds so strongly, it switches the entire enzyme completely from the T state to the R state. So what we're seeing on the right there is ATCase bound to pala, which we can see here. So we've switched completely from the T state to the R state. This has high affinity for its substrate, even higher affinity for pala because it binds the active site so well. Now, ATCase, like any allosteric enzyme, exists in equilibrium between the T state and the R state. So we can consider that we have T to R, there is some equilibrium between them. The equilibrium between them is not an equilibrium constant in terms of K that we talked about before, but instead is defined as L, the allosteric coefficient. That's simply the ratio of the concentration of enzyme in the T state to the ratio of concentration of enzyme in the R state. Okay? So, here, the T state has low affinity for the substrate, has low catalytic activity, so it's inactive, and is favored in the absence of substrate. So if we come back over here, isolated ATCase, right, on its own, is going to be mostly in the T state by a ratio of a couple hundred to 1, as we'll see in just a minute here. The R state has higher affinity for the substrate, is in the most active form, and is favored upon binding of the substrate. So when the substrates bind to the T state, at first it will happen slowly in terms of ATCase due to its cooperativity, but eventually it'll bind at all 6 and will adopt the R state. So similar again to what we saw for hemoglobin, right? We have the R state curve in the blue, we have the T state curve in the red. And this makes sense, of course, at any given concentration of, say, in this case, aspartate, if we hold the concentration of carbon monophosphate steady, right? Now, as we add more aspartate, at any given concentration of aspartate, the activity of the R state is higher than the activity of the T state, right? The sigmoidal curve for an allosterically controlled or cooperative enzyme comes about because of the superposition or the what's the word here? Did I write it down? The composite, right? This is the composite of the R state and the T state. If we take those two functions and composite them together, we get a sigmoidal curve. Okay? So the substrate itself, its cooperativity, what generates the sigmoidal D naught curve that we see here. Okay? Cooperativity generates the sigmoidal curve. And this is called the homotropic effect, right? A homotropic effect because homo is the same enzyme, it's the active site of an enzyme affecting the binding of substrate at other sites in the same enzyme. Homotropic. Homotropic effects lead to this sigmoidal curve. Now, why does this sigmoidal curve matter, right? Well, first, the transition occurs within a narrow range of substrate, okay? And we get a threshold effect. So what does this mean? Well, here we have two hypothetical enzymes. Okay, two hypothetical enzymes. And what we'll see, right, at near Km, we get, for the red, no line here. For the allosterically regulated enzyme, we look at the Km, which is somewhere around here, okay? Well, now, within a small range around Km, we get a very steep increase or decrease in activity, and compare that with the non-allosterically controlled enzyme. So let's just take a hypothetical situation. We want to increase the rate, right, from 0.1 Vmax to 0.8 Vmax. So we want to increase the activity from 10% all the way up to 80%. Well, if we do that, you see that we need 27 times more S to reach 80% compared to what we needed to reach 10%. So we need a 27-fold increase in substrate concentration. If we have an allosterically controlled enzyme to reach that same Vmax, okay, we only need a 4-fold increase in substrate concentration. So it allows an acute response to changes in substrate concentration, which is useful for these enzymes. We want to be able to quickly and acutely respond to changes in the amount of substrate available. Okay? So, suppose a mutation in ATCase decreases an enzyme's Km, right? Would this mutation push ATCase towards the T state or the R state? And what would that graph resemble? Someone who has an answer to it.
The R state.
Huh?
The R state.
The R state. Why?
And it would shift leftwards.
Because it would shift leftwards, right? We can look at this mathematically. If we have the function for an allosterically controlled enzyme here, right, with a Km of 26, whatever, millimolar, right? Well, if we increase the Km, right, we shift to the right, okay? We need now 76 millimolar to reach 0.5 of V naught, right? 100% V naught or V max is at 1, so to reach 0.5 we need 76 millimolar. If we decrease from 26, well now we need less and less and less and less until we resemble what's functionally just a Michaelis-Menten enzyme, right? And this is what the R state is. It's essentially resembling or mimicking a Michaelis-Menten enzyme. Right? We're shifting the Km. Alright, so what happens here? When ATCase then binds CTP, which is not a substrate, we call the substrate for ATCase are carbamoyl phosphate and aspartate. So when ATCase binds CTP at its regulatory site, at its allosteric site, within the regulatory subunits, what happens? Well, what do we think is going to happen? Okay? Well, we saw before that CTP decreases the rate of the reaction, right? The more CTP we have, the rate of the reaction decreases. So what do we think that CTP is doing in terms of the T state and the R state?
Stabilizing the T state.
Exactly, stabilizing the T state. Remember, the T state is the less active of the two states. Okay, so if we want to decrease the rate of the reaction, we want more T state, right? So we can categorize that or quantify that rather by looking at the allosteric coefficient under different concentrations of an allosteric allosteric coefficient of ATCase in the absence of CTP, we see that it's got a T to R ratio, or L, of about 200. Now, when we add CTP, what happens? Well, that allosteric coefficient increases now to 1,200, right? Or what is it, 1250? Okay? So what does that mean? Well, let's remember what L is. L is simply just the ratio of T to R. Here, right? L is the ratio of T to R, so the higher the L, the more T we have. So a negative allosteric regulator, or an allosteric inhibitor, will increase T, whereas a allosteric activator, okay, will decrease L, or increase R, in other words, decrease L, okay? Allosteric regulators can be positive or negative, so CTP is an example of an allosteric inhibitor. We can have allosteric activators, like in this case ATP, right? ATP, you see here, is an allosteric activator of ATCase. In the black, what we have is the V0 curve in the absence of either ATP or CTP, right? When we add CTP, as we imagine, at any given concentration of aspartate, again, assuming that we're holding the carbamoyl phosphate concentration stable, all right? What we see is that at any given concentration, the rate of the reaction is lower in the presence of CTP, but at any given concentration, the rate of the reaction is higher in the presence of ATP, right? So you can have an allosteric activator or an allosteric inhibitor. Okay? So these heterotropic effects, okay, different than homotropic, are effects on the kinetics of an enzyme due to a molecule or effector that is not the substrate. And what these do is that they increase or decrease Km, right? So we saw that before with our little graph. The effect of having an allosteric activator or inhibitor, right? So an allosteric inhibitor, like CTP, is going to increase Km, right? Shifting that curve to the right, so it resembles a T state. An allosteric activator will shift it to the right, so it resembles the R state. So it shifts the Km, and they impact the activity again by altering the coefficient of the allosteric coefficient. All right, so why do we think that CTP acts as an inhibitor of ATCase, while ATP acts as an activator of ATCase? There's a very specific reason that makes sense here. Right? Why do we want those to act together? Say that again, say it loud.
There's enough product being formed, doesn't need more to be produced, so ATP acts as a inhibitor for ACPA.
Okay, so let's break that down. If there's product being formed, then we don't need to make any more product, right?
There's enough.
There's enough, right? So when do we know that it's enough? Say it louder.
When CTP is able to outcompete ATP.
No, not to do with competition. When is the level of CTP going to be high? Let's start there. If CTP is high, then what's going on with CTP? It is going to inhibit it, right? But just backing up from there, if the concentration of CTP is high, then what's being done with CTP right now in the cell?
It's not being used.
Yeah, nothing. It's not being used. It's building up, right? So, to your point, yes, if the concentration of CTP is high, that means it's not being used, it means that it's not needed, okay? So let's turn off the committed step here so that all the precursors, carbamoyl phosphate and aspartate, they can be used for other things. We can free those resources up. Now, why is ATP an activator of ATCase? Where's Angeles, Pinas? You here today? All right. Oh, yes. All right, ATP ATP is a form of energy, right? So we have a lot of ATP. That means, as we'll see later on when we talk about metabolism, the energy charge of the cell is high. That is correct. But what does that mean that we need more CTP? You have your hand raised? Yeah, okay. Sure. ATP is going to be necessary. We'll see, hopefully, if we get to it. ATP is necessary for generating parts of CTP, right? But there are other mechanisms that deal with maintaining the energy charge of the cell, right? But let's think about this. Okay, so we have CTP, which is an allosteric inhibitor, and we have ATP, which is an allosteric activator. Okay? So let's say that ATP is present and it's able to outcompete CTP for binding to the regulatory site, so we get a net activation of ATCase, right? What does that tell you about the levels of ATP and CTP? Which one is higher? ATP, right? Why then, if the level of ATP is higher than the level of CTP, why do we want to increase the amount of CTP that's being made? It has nothing to do with energy. Four. Right. If we have an abundance of ATP compared to CTP, that means that enough ATP is available for synthesizing either RNA or DNA, more than we have CTP available to synthesize RNA and DNA. And because all four nucleotides are present in DNA and RNA in roughly the same amount, or right about equimolar, right? Well, if we have too much ATP compared to CTP, then we need to make more CTP so we have this balance, right? As all things should be. So moving on to isozymes. Isozymes, so that's allosteric regulation. Isozymes is another way that we have to regulate the activity of enzymes. But this works a little bit differently. We're not directly regulating the enzyme itself. Instead, we're regulating different versions of the enzyme in different tissues. Okay? So isozymes are essentially just that, different versions of the same protein. So we can have isozymic versions of, say, lactate dehydrogenase, we'll talk about, or we have isozymic versions of glycogen phosphorylase. Okay? And these are present in different tissues because different tissues have to do different things, obviously. Otherwise, there would be no difference or no purpose to tissue differentiation. Right? So these are coded for by different genes. Right? So say that we have a muscle isoform and we have a liver isoform of an enzyme. Right? A muscle isoform and a liver isoform. The muscle one is turned on in the muscle and turned off in the liver, whereas the liver isoform gene is turned on in the liver and off in the muscle. So we get expression of what is functionally the same protein, all right, but with different kinetic and regulatory parameters, right? So they behave differently, they respond to different things, they catalyze reactions at different rates. Okay, this is important again specifically for liver isoforms compared to, say, skeletal muscle isoforms. And these genes are again activated in different tissues. We can also activate these isoforms at different developmental stages, right? So in addition to being present in different tissues, we can turn on or off different isoforms during different developmental stages. What's an example of that we've talked about already? Hemoglobin, not myoglobin. Myoglobin is always expressed in the muscles, and hemoglobin delivers oxygen to the muscles and is taken up by myoglobin. Adult hemoglobin and fetal hemoglobin, right? The gamma chain, which we find in HbF, is essentially an isoform of beta globin, which we find in HbA, right? They do the same reaction. They're binding oxygen they do so in a different way, right? They're regulated differently. Remember, how does the gamma chain function to increase the affinity of fetal hemoglobin for oxygen? Well, it decreases the affinity of HbF for 2,3-DPG, right? So it's got a different regulatory strategy or parameter than does HbA, right? So that's an example isoform. So here, for example, an isozyme of lactate dehydrogenase. Lactate dehydrogenase is tetrameric, meaning it's composed of 4 identical subunits, right? So this tetrameric protein that catalyzes a step in anaerobic glucose metabolism, right, and glucose synthesis. We'll talk about this in chapter 16, but essentially the reaction that it's catalyzing is the oxidation of nicotinamide adenine dinucleotide, NADH, being oxidized into NAD+, and pyruvate is being reduced to lactate, right? This is lactate fermentation. This is the end product, essentially, of anaerobic glycolysis, right? So in the absence of oxygen, this is what happens. The pyruvate that we form from a glucose is reduced to a lactate, and that releases lactic acid, right? And there we go. Now, humans have two isoforms of, humans have two isozymic polypeptide chains for LDH, right? The H protein is highly expressed in the heart muscle, right? So H for heart. The M is expressed in skeletal muscle. Okay, so again, think about what's going on here. Different tissues, heart and the muscle, they have different functions. Okay? The muscle is responsible for moving our limbs, occasionally, right? So they quickly increase in the amount of energy they need, but then at rest, they kind of slow down, they don't need as much energy. Our heart needs constant energy, right? Our heart, hopefully, never stops, right? It stops exactly once, right? So we need a constant supply of energy. So anything to do with metabolism is going to vary drastically in our heart compared to basically any other tissue, right? So we can see that here. Okay, here we have different isoforms of lactate dehydrogenase, right? So the circles represent, give me a sec here, the circles represent the heart isoform, the H isoform, and the squares are representing the M isoform, right? Now the M isoforms have a lower affinity, right? So they are more effective when concentration of oxygen is lower, okay? So the heart has a lot of oxygen, right? The heart is highly oxygenated, so they express the H isoform, while the M isoforms, in the muscle, there's a lower PO2 of oxygen, right? So they express the M isoform, right? So the H isoforms are more effective when PO2 is high, and the M isoforms are more effective when PO2 is high. Low, right? So heart works better at high PO2, whereas M works better at low PO2, right? And the M is being represented by the circles, and the H is being represented by the squares, right? So if we look at the top there, what we're seeing is, it's kind of gross, right? But looking at fetal mice or fetal rats, okay? And they examined the proportion of lactate dehydrogenase being expressed in the heart at that time, okay? So what's going on here? Days before birth, so 9 days before birth, 5 days before birth, and so on. In the little fetal mouse, all right, we're expressing mostly the circles, and the circles again are the M isoform, right? We're expressing the M isoform. Then, closer to birth, we're expressing mostly the M isoform with a little bit of the H isoform. At 1 day before birth, a little bit more H. 12 days after birth, mostly H, and 21 days after birth, mostly or all H, right? So why does this make sense? Okay? The mouse is expressing in the heart, in the fetal heart, expressing lactate dehydrogenase that would normally be expressed in the muscle. Why would it need to do this? And famously, what don't fetuses do?
It's because fetuses don't have a lot of oxygen.
Huh?
It's because of the lack of oxygen.
Yeah, right? They're still in the womb, right? So they don't have access to high partial pressures of oxygen. So instead, they need to express in their heart right now an isoform that is more efficient, at, or rather, is more efficient at low oxygen concentrations. Now, as it grows, right, as it progresses through its developmental stages, now in the heart, the amount of M goes down while the amount of H goes up because now it needs a lactate dehydrogenase that is more efficient at high partial pressures, right? So we can regulate both in terms of developmental stage, which isoform is being produced in order to meet the needs at that developmental stage, or in a tissue-specific manner, as we'll see as we go on through this message. Now, covalent modifications are critically important in regulation of enzymes, right? And specifically in metabolic enzymes, right? So we're done with isozymes, now we're moving on to covalent modifications, right? And what is a covalent modification? Well, basically the functional groups of several residues can be covalently modified to alter its properties and affect the protein's structure or function, right? So examples of covalent modification could be phosphorylation, and critically important for metabolism. Could be acetylation, like we see here with an acetylated lysine. Anybody know really where we see a lot of acetylation? Histones, right? Regulating the chromatin structure. And glycosylation, right? Glycosylation is interesting on its own. We're going to talk a little bit about it in chapter 11. Sugar modifications, right? The reason they're so interesting is that they're kind of a third genetic language, right? So we have the genetic language of nucleotides, the genetic language of amino acids. Sugars constitute a third genetic language, right? Or biological language. And it's still being deciphered. We still don't know exactly what it is. Why it's so interesting is that a residue can be modified by one sugar, but that sugar can be linked to another sugar, which can be linked to another. Some sugars can be linked to two sugars, right? So we get these branching structures that essentially modify a protein called glycoprotein. What these sugar modifications do, right, is easy to determine once you know that it's modified, but predicting what they do based on the glycosylation structure is still not completely understood. Again, we'll talk about that in chapter 11, right? Now, what's important for covalent modifications like allosteric regulation and isozymic regulation in terms of development is that they're reversible, right? Allosteric regulation is reversible by dissociating the allosteric regulator from the enzyme. Isozymic regulation is, at least in terms of the developmental stage, can be reversible by switching on and off a gene. That doesn't really happen. But here, covalent modification is easily reversible, right? So it's a good way to turn on and off a protein. Now, the most important covalent modification, which is also called a post-translational modification, since the protein is modified post-translation, in metabolism, again, is phosphorylation. Phosphorylation is catalyzed by a family of enzymes called kinases, right? They belong to the enzymatic class that we talked about in chapter something, 5, I think, transferases, right? So they're transferring a phosphoryl group. And where they're transferring that phosphoryl group is almost always ATP. And what they're transferring onto is any residue that ends in a hydroxyl, namely serine, threonine, and tyrosine. Tyrosine kinases are kind of their own thing, right? They have very specific functions we'll talk about. Actually, you'll talk about some of them with Dr. Barber. By the way, chapters 13 and 14, Dr. Annika Barber is actually going to be doing those for various reasons. So you'll meet her in a couple of weeks after the exam. Either way, so you'll talk about tyrosine kinases with her, I think. And basically what we have is this. So here what we have is a serine, right? So a serine and threonine kinases belong to the same thing. And what we have, again, these end in a hydroxyl, you're looking at a serine. The gamma phosphoryl group, which is to say the farthest phosphoryl group in ATP, is transferred from ATP onto the hydroxyl, generating, in this case, phosphoserine or phosphothreonine and ADP, right? Only intracellular proteins are modified by phosphorylation, right? Only intracellular proteins are modified by phosphorylation, and why is that? Yeah, because you need ATP, and there's no reason why a cell would export ATP. So there is essentially zero ATP outside the cell. So extracellular proteins cannot be phosphorylated. Now, kinases work on this concept of consensus sequence, right? Many enzymes work on a consensus sequence. binding enzymes, kinases, right? What is a consensus sequence? So if we look at, for example, protein kinase A, pKa for short, okay? It's got a consensus sequence here, RRXSZ or RRXTZ, where S and T are the serine and threonine to be phosphorylated by protein kinase A. So what does this mean, right? Well, this is the sequence that is going to be recognized by protein kinase A. An arginine, followed by X, which is a small residue, like an alanine, followed by a serine or threonine, which is going to be phosphorylated, followed by Z, which is the large hydrophobic residue. Okay? And how did we figure this out? How would you figure it out? How would you determine what the consensus sequence for protein kinase A is? Say that you don't know what it is. What experiment could you do? Imagine that you could do any crazy experiment in your head. How would you figure this out?
Change the amino acid sequence.
But how would you know which amino acids to change? Huh? Right, but you don't know the sequence yet. We're trying to figure out this sequence.
Get access to PKA of different species, you look at what's conserved across the.
Alright, so if you had access to pKa of different species, you see what's conserved. But this is the sequence that's being recognized on the substrate. This sequence is not in pKa, right? So the way that we figure out consensus sequences is essentially, right, we have a population of proteins that we know are targets of pKa, right? They're substrates of pKa, okay? We can figure out the sequence for those proteins. Now we have the entire sequence. We now mix those proteins with pKa, okay? And we see which residues are phosphorylated, right? We know the sequence of the substrates, right? That's easy enough to do. We'll talk about that at the end of the semester if we have time. But we can just as easily look at the sequence and then try to see which ones are phosphorylated. So we'll see, for example, in a hypothetical protein that S at position 148 is phosphorylated. So this is substrate 1 of pKa. Substrate 2 of pKa. We'll see that we've got phosphorylation at S47. Substrate 3, we'll look and we'll see that we have phosphorylation on T205, let's say, right? So what we do then is we just look at, okay, let's take the surrounding 10 amino acids or residues from here. So we'll take 10 that way and 10 this way. And we'll take 10 this way and 10 this way, 10 this way and 10 this way. Right? Take all these up. We line up all the serines and threonines together, right? And we see, all right, at the position right before the serine, which is the amino acid that we see the most? At 2 amino acids before, what's the position, what's the residue that we see the most? And so on and so on. We can do after and after, right? So what we see is that at 2 positions after the serine or threonine, the identity of the residue is random. It doesn't matter. But at 1 position after the serine or threonine, what we see is that we have to have a large hydrophobic residue. Then we have the serine or threonine. 1 position before the serine or threonine, we see that we have to have any small residue. Sometimes it's a glycine, sometimes it's an alanine, right? 2 positions before and 3 positions before, we see that those are always arginines, okay? And then 4 positions away and 5 positions away, we see that the identity now no longer matters. So we see, all right, well, we have a pattern now. We have mostly arginines at position -3, mostly arginines at position -2, mostly nonpolar residues large at position -1, our position 0, which is serine or threonine, and then mostly large hydrophobic residues at position +1. Right, so there's our consensus sequence. Any kinase is gonna, or serine threonine kinase is gonna recognize obviously a serine or threonine. And how does it know which serine or threonine to phosphorylate? It can't just recognize any serine and threonine. It has to be a specific one for the reason that we'll see in a minute. Okay? So the way it does that is by looking at the residues surrounding it, right? Forming weak interactions with the residues around it, making sure to put the correct serine or threonine in the active site so that it can transfer the phosphoryl group from ATP onto that serine or threonine. Oops. All right. Protein phosphatases reverse that reaction, right? So phosphorylation of a protein, so a serine or threonine, is irreversible. Okay, we cannot go backwards there. Phosphatases allow us to go backwards. So a protein, you might imagine, can be turned on by phosphorylation, right? So a protein is recognized, let's say, by protein kinase A, that our substrate now, our metabolic protein, has the consensus site for protein kinase A is phosphorylated and is now turned on. Great, so it was off before, and now it's on, so we've turned it on. But what happens after that metabolic signal is gone, right? So the glucagon receptor turns off, or the adrenaline receptor turns off, whatever. That signal's now no longer there, but we've already turned on our metabolic protein. Well, now the signal is gone, so we need to turn that back off. And this is where protein phosphatases come in, right? So protein phosphatases essentially just remove phosphoryl group. They remove it via hydrolysis, right? So we have our phosphoprotein, in this case, let's assume that's a phosphoserine, right? Plus water yields what we call orthophosphate, plus now our just regular serine, okay? So, let's write a balanced reaction for the phosphorylation of an enzyme E followed by its dephosphorylation. What's the ultimate result here? Okay? What do we think? Anybody see the eraser? Shut up. All right. Okay, so we have an enzyme. What else do we need for phosphorylation? Yields what? Huh? ADP. What else? Right, our phosphorylated enzyme. Now for a phosphatase, what do we write down first? What is a phosphatase going to do? It's going to remove a phosphorylated, or a phosphoryl group rather, so we need our phosphorylated enzyme. What else do we need? Water. What's the product here? What's one product? Enzyme plus, yeah, an organic phosphate or orthophosphate. Okay? So a balanced reaction is essentially just canceling out things that appear on both sides of the arrow, right? So here, this cancels out, the E cancels out, so our balanced reaction is ATP plus H2O yields ADP plus orthophosphate. This is the reaction for ATP hydrolysis, right? So this cycle, right, if these were to happen at the same time, the balanced reaction is simply ATP plus H2O yields ADP, right? So why don't we want to do this, right? Why is it useful that phosphorylation is regulated by kinases and phosphatases? What would be the result if it weren't regulated? Waste of energy, exactly. We just be burning through ATP for no reason at all, right? So it's good that these are regulated. You need a kinase to add a phosphoryl group and a phosphatase to remove it, and those kinases and phosphatases are themselves regulated, right? So how does phosphorylation affect enzyme activity? Most importantly, right, the free energy of phosphorylation is large, right? The standard free energy for ATP hydrolysis, this reaction here, the standard free energy is negative 30.5 kilojoules per mole in the cell because the concentration of ATP is high and the concentration of orthophosphate is low. The actual change in free energy in the cell is closer to 50 kilojoules per mole. Right? About 25 kilojoules of that are used for the actual phosphorylation, right, which makes this process irreversible. The other 25 kilojoules go where? Right? We're we're releasing 50 kilojoules per mole when we hydrolyze ATP to put the phosphoryl group onto the enzyme, right? We're releasing 50 kilojoules per mole. 25% of that only, or rather 50% of that only, is used to actually transfer the phosphoryl group. So what happens to the other 50%, the other 25 kilojoules? Well, what has to happen to it first? Well, it has to go somewhere, right? What's one way that it could go? It could be released as ATP, right? What's the problem with that? We release too much heat, right? So we can't do that. So where's all that extra energy going? That extra energy is being stored in the enzyme, right? And that enzyme can do things with it. It can use that energy to change its conformation, right? It can use that energy to do work, right? It can use that energy to do other things. So the energy of transferring the phosphoryl group onto the enzyme actually gives that enzyme the ability to change itself, right? By providing that energy. A phosphoryl group adds two negative charges from the phosphate, okay? And this can form sulfurates, can form ionic interactions, can help stabilize certain interactions between the protein, so it can change the protein. Phosphoryl group can form 3 hydrogen bonds, so similar to the point before, right? It can form interactions stabilizing certain conformation of the protein. It might be active or inactive. The kinetics of phosphorylation and dephosphorylation can be adjusted to meet the timing needs of a particular process, right? So phosphorylation itself can act as a timer, as we'll see when we talk about the G protein coupled receptor cascade, the GPCR cascade, right? G alpha, all right? Again, don't get bogged down with this, but G alpha is a G protein that binds GTP, right? It's got phosphatase activity, or it's got GTPase activity. It can hydrolyze GTP, similar to what a kinase can do, but it does so very slowly, right? So phosphorylation and dephosphorylation can occur over a matter of seconds or milliseconds, or it can occur over a time frame of minutes, right? So it can act as a timer. Phosphorylation can be used to amplify signals, as we'll see again when we talk about signaling cascades. One protein kinase A, for example, can phosphorylate many hundreds or thousands of proteins, right? So one protein kinase A can lead to the activation of thousands of proteins. That's an amplification of the signal. And ATP is the cellular energy currency, so this also allows phosphorylation to serve as an energy sensor, right? The more ATP there is, the more phosphorylation can occur, and that can tell different proteins different things, right? It can tell, if ATP is high, then phosphorylation is abundant. We might phosphorylate a metabolic protein, inactivating it, meaning that it's not going to generate as much ATP because the ATP is already high, right? Not going to talk about activation of protein kinase A because it's kind of irrelevant right now, and we are going to talk about it when we talk about something else, so that's fine. When we talk about signaling cascades, we talk about protein kinase A. So we will see this figure again in chapter 12, 13, or 14. I can't remember which right now. Some enzymes are also activated by proteolysis, right? I don't spend too much time on this, okay? But the folded forms of some enzymes are inactive until one or multiple specific peptide bonds are cleaved, right? Meaning that when this particular protein is synthesized, it doesn't do anything. It just kind of hangs out there inactive, okay? These are called zymogens, and these typically end in ogen, or they begin with pro, right? So we can have prothrombin or chymotrypsinogen, as we'll see in this film. We're just going to talk about chymotrypsinogen. Proteolytic cleavage does not require an energy source and is irreversible, okay? And what does this mean? Remember that the delta G naught prime, the standard free energy change for the hydrolysis of peptide bonding between -15 and -20 kilojoules per mole. So it will happen spontaneously, but we talked about in chapter 5 how this doesn't happen because the activation energy is quite high unless we have a catalyst or an enzyme. And that's what we have in the form of a protease. The protease simply increases the rate of that reaction. It increases the rate of peptide bond hydrolysis, and specifically because it's an enzyme, it can specify which peptide bond it's going to hydrolyze by recognizing the residues on either side of the peptide bonds. This plays a role in several processes, specifically digestion and blood clotting. We're not going to talk about blood clotting right now because that's going to haunt you in your dreams enough if you go to medical school. So we are going to talk about digestive enzymes here a little bit. And, all right, so that's fine. So basically, we can keep an enzyme off until we're ready to use it, right? By exposing it to a specific protease, that zymogen then becomes activated. So let's look at chymotrypsin. Chymotrypsin is the digestive enzyme that cleaves peptide bonds after residues with large nonpolar R groups. Okay? Synthesized as chymotrypsinogen, which is inactive, is synthesized in the acinar pancreatic cells. They're stored in granules and then released into the duodenum when the cell is told, so either by a hormone or by a medium. And you can see that in the figure on the right here. Okay, we have synthesis of our zymogen, or our chymotrypsinogen, into the rough ER, which then is trafficked to the Golgi. It's packaged into these granules, right? So what we have here is a zymogen granule, which contains, of course, chymotrypsinogen, but other zymogens as well, right? Here, once the signal is received, either a hormone or a nerve impulse, this zymogen granule, which contains all of these digestive zymogens, fuses with the plasma membrane of the acinar cell and is released into the duodenum of the intestine, right? And now we have all of these zymogens out here. Okay. Why do we want to do this? Why do we want to synthesize these digestive proteases as zymogens, right? So these digestive proteases, which are meant to digest our dietary proteins, are synthesized as zymogens. Why is that important to do? Sure.
Because the entire body is made out of protein and it will digest all of it.
Yeah, exactly. So if we have, right, our chymotrypsin or other dietary proteases being synthesized active, what would happen? They'd just start chewing up all the proteins in our cells. So we want to keep them inactive until they're released from the cell into the intestine, ready, right? The duodenum is right where the stomach empties into, right? So right there, we want to activate so that they're inactive with the cell and active as soon as they get a dietary protein. And how is chymotrypsinogen activated? Well, cleavage of chymotrypsinogen by trypsin at a couple of positions activates it, right? So we have chymotrypsinogen. This is cleaved by trypsin, right? Here, between positions 15 and 16, okay? This generates this high chymotrypsin. High chymotrypsin is active, okay, but it's unstable. But it is able to actually, to cleave peptide bonds as chymotrypsin typically does. But this pi chymotrypsin does something interesting. Pi chymotrypsin acts on other pi chymotrypsin molecules to cut two more places. It cuts here, right, and we lose two dead peptides, 14 and 15 on this side, and 147 and 148 on this side, right? This is now alpha chymotrypsin, which is active. But why does going from chymotrypsinogen to pi chymotrypsin, how does that activate it? Well, it frees up this here position at lysine, isoleucine 16 rather. Here, this the N terminus of isoleucine 16 is able to rotate, right? So here we have chymotrypsinogen. After cleavage, the N terminus of isoleucine 16 over here twists around, forms an ionic interaction with a aspartate 194, and this completes the specificity pocket. In other words, it completes the active site of chymotrypsin. So before cleavage, the active site is incompletely formed and it can't recognize the peptide bonds that are supposed to be cleaving. After trypsin cleavage, after trypsin cleavage, right? Now that isoleucine 16 is able to twist around and bind with aspartate 194, and this completes the active site so that it can bind its substrates, right? Now, where does this trypsin come from? Trypsin comes from trypsinogen, right? So trypsin is also synthesized as a zymogen. And trypsin is activated by enteropeptidase, right? And this is an example of, essentially, this is an enzymatic cascade. Enteropeptidase is present on the cell surface of cells that line the duodenum. These cells here, right? So here's the lumen of the intestine. Here's the cytoplasm of these acidyl cells. On the cell surface here, we have this enteropeptidase, right? These are always active. And enteropeptidase is going to act on trypsinogen, that zymogen, and it's going to cleave it to activate the trypsinogen, turning it into trypsin. Okay? Now this trypsin that's formed by enteropeptidase is going to act on other trypsinogen molecules, activating them, as well as activating chymotrypsinogen, proelastase, procarboxypeptidase, and prolipase. Prolipase is not a protease. It's something that breaks down fatty acids. We'll talk about that later on. So ignore this for now. But trypsin, right, activates more trypsin and activates all these other peptidases. And all these other peptidases then go on and start to digest our dietary proteins, right? So from one action here, right, the activation of trypsinogen by enteropeptidase, you get the activation of many downstream proteases which can now go on and digest our dietary proteins, right? So, trypsin cleaves proteins from the carboxy side of lysine residues. Trypsin has a specific inhibitor, right, called the trypsin, or the trypsin inhibitor, pancreatic trypsin inhibitor, okay, that binds to trypsin particularly well, right? Trypsin inhibitor has a lysine residue though, that binds to trypsin but is not hydrolyzed. Why is this the case, right? So what are we talking about here? Right? Recall that trypsin cleaves after lysines and arginines, right? Trypsin cleaves after lysines and arginines. But in the pancreas, right, there's expressed a trypsin inhibitor that binds to trypsin and prevents it from hydrolyzing peptide bonds after a lysine or arginine. But the trypsin inhibitor itself has a lysine that should be degraded by trypsin, but it's not. All it does is inhibit trypsin. Why do you think that might be the case? There's a lysine residue on the inhibitor peptide that binds to the active site but is not hydrolyzed. You go back here, right? A hint is to notice something about the structure of the trypsin inhibitor. Do you notice any change between free inhibitor and bound inhibitor? Do you see any change here? In the back, you're shaking your head. You don't see any difference, right? Like there is no difference. So what does that tell you then about this binding? Do you think it's strong or is it weak? It's strong, right? Because there is no change in the structure. That means that energy didn't have to put in to change the structure of the inhibitor in order to fit the active site better. So that means that this substrate binds very well to the active site, right? So why would that lead to inhibition? This goes back to chapter 5.
It's a better competitor than the other lysine salaries.
It's what?
It competes better than other lysine that would try to.
It competes better?
Yeah.
It competes better. All right, well, sure, but it's a lysine residue, so why isn't the peptide bond after the lysine getting hydrolyzed?
Kind of being like induced more like long-term stay there, stay pop.
All right, remember, there's the best binding energy between an enzyme and the transition state. But in this case, the structure of the inhibitor is such that the binding energy is now maximized between the inhibitor and the enzyme. So that now going to the transition state requires the activation energy plus the binding energy, right? So in this case, it stabilizes the ES complex, preventing it from ever reaching the enzyme transition state complex, right? Let's see. I want to talk about gout. And this. All right, if there's time on, what's today? If there's time, we'll go over this on Tuesday. I will post, so I'm gonna take pictures. I've got too much going on. I will try my best to post these directly after lecture before I leave.